Alright, so this lecture is all about mutations, specifically gene mutations that occur within DNA. So sometimes during replication or when exposed to something called a mutagen, mutations are going to occur. And a mutation is just a change that occurs. You guys know that DNA is composed of adenine, guanine, thymine, and cytosine, or all those A's, T's, G's, and C's. And sometimes an A gets changed to a C, or a G gets changed to a T. So it's just any change that occurs within that code of an organism. So sometimes when you replicate your DNA, the parts don't match up. So sometimes an A will bond to a G, and we know that that's not how it's supposed to be, right? An A bonds to a T. But sometimes you have a lot of DNA and mistakes are going to be made. Your body's not perfect. So sometimes a mutation occurs there, and other times it's is when it's exposed to something called a mutagen. And a mutagen is just something that's going to increase the likelihood of a mutation. Often it's things like radiation or pollutants, chemicals, or certain microorganisms like viruses and bacteria. So this is a picture of sun exposure. So you see this picture under normal light, and I believe this is of UV light, and you can see all of the little sunspots that are kind of like freckles. Freckles are created because of increased sun exposure, but you can see here, even though that we cannot really see them under natural light, you can still see that her skin is damaged. Here's also a picture of how many cancer fatalities. So cancer, we know, is often uncontrolled cell growth, which is often because of a mutation that occurs in a certain gene that is really important for regulation of the cell cycle, or mitosis. So tobacco, air pollution, and other nasty things can sometimes lead to mutations that can lead to cancer. And this is a picture of human papillomavirus, or HPV, which is known to cause cervical cancer. So ladies, and maybe even gentlemen, I don't remember what the government really says as far as recommendations, but you guys probably received an HPV vaccine that will help decrease your risk of cervical cancer. So sometimes the environment, chemicals, and even some microorganisms can cause your DNA to mutate. So when they occur in somatic cells, they can only affect the tissues of that organism. So remember that somatic cells are body cells, okay? Eye cells, lung cells, toe cells, eye cells, heart cells, muscle cells, all those types of cells. They are going to be only that tissue. So when would that have occurred? Meiosis or mitosis? I'm going to give you like 2 seconds to think. Okay, remember that. Somatic cells, mitosis. Okay, so that's like if you have a lot of sun exposure and you never put sunscreen on your face, you will get maybe a mutation in a skin cell that's on your nose, and you might get melanoma or skin cancer just on that tissue that was exposed to the sun. Other times, that mutation can occur in a sex cell, or egg and sperm, and may be passed on to offspring without affecting the parent. So that's like when the parents are perfectly normal, but their child has a genetic disorder. So during what process would that happen? It's meiosis, making those egg and the sperm. So that's how we get those genetic diseases, which is what your project is about. So, question, are all mutations bad? Well, I know it kind of leaves a yucky taste in our mouth. We think mutations are a bad thing, but they're not. Okay, so this is a picture showing you the skin pigmentation based off of race. So you can see at equatorial regions, you have darker skin tone, and that's because the sun is more intense. Versus if you are up far north or far south, you're going to have a lesser skin tone, and that's because of natural selection. So if you take genetics with me, Miss Mueller, I'm going to teach you all about the genetics of skin color, but that is one reason why. If we didn't have mutations, we would all be this color, because we're thinking that Homo sapiens, or our species, originated in Africa. So if you want to learn more, talk to me. I'll give you some resources. So, and that's also the reason why we look so different, okay? The reason why our noses are the way they are, our hair color is the way that it is, our eye color, our face shape, tall, short, all of that is because of mutations. So mutations, Not always a bad thing, but they definitely can be. So something called a central dogma is just the central focal point of life. And that is DNA going to RNA, going to protein, which leads to a trait. So remember, when that protein gets expressed or when it's being made, it's going to lead to some characteristic that often we can see, such as hair color. So proteins are what put the instructions of DNA into action. This is just an instruction manual. This is the output of those instructions. So again, DNA goes to RNA, goes to protein, and there's those ever important words, transcription and translation. So here are a lot of the functions of proteins, but you guys have already done something in a station. But if you want, pause the slide and you can look at all of these different proteins that occur within our body. So if the DNA code is changed, what's going to happen to the protein and therefore the trait? So think about that. I'll give you like 2 seconds. So if I change the DNA code, it's going to change the protein and therefore the trait. So you guys are also going to be doing something about sickle cell anemia. So if I change my DNA, then that's going to change the protein, such as something called hemoglobin. Okay, hemoglobin is something that helps bond to oxygen and help us deliver oxygen to our tissues. You're going to have a change in a trait, or what you're going to learn next unit, something called a phenotype. So if I change the DNA, I'm going to change what things look or act like. So there are several types of mutations. Okay, 2 main groups are called point mutations and frame shift mutations, and I'll go through each one. You should have a table in your notebook, so make sure you're good to go, okay? So a point mutation is when a single base pair is changed. So in this picture, an A gets changed to a T. So a type of a point mutation is called a silent mutation, in which the amino acid does not change. So if you look at a codon wheel, CAA codes for valine, CAT also codes for valine. So even though I changed my DNA code, I didn't get a net effect. I still have valine. So silent mutations is when something is changed, but we don't really have an overall effect when it comes to protein function. A missense mutation, it results in a different amino acid, but the protein is still mostly the same. So here I changed a G to a T, which changed the, I believe this is tryptophan, to cysteine. Okay, so missense mutation is a slight change. Nonsense is when you have a premature stop codon. So here, instead of having TGG for tryptophan, if I change the G to an A, that leads to a stop codon. So when you think about it, if this had so many more codons and it was like a bazillion more amino acids, that would be truncated and it would be 2. So when you think about it, how well is this protein going to work? Think about it. Frame shift mutations is another part. So that is when the change in the reading frame occurs, because remember, amino acids are read in 3s, so the codon sequence. So insertion is when I just stick a base pair in, okay? I just insert something. So here's an example. Instead of a T and then a G, I inserted a C. So that is going to move this G over and that G over to the next codon and so on and so forth. So everything's going to be changing after that insertion. So think about it. How well is this protein going to be working as opposed to this protein? Is it going to be having the same function? Sorry, there was an announcement over the intercom. Continuing, insertion. Okay, insert a base. That kind of screws everything up. Okay, sorry about that. Okay, then there's deletion. Okay, so that is like this T was deleted, which moves this G over, this C over, this C over, this G over, and everything down the line that I'm not showing. So like the T got removed. So again, the whole reading frame is going to be screwed up. This amino acid is going to be the same, but everything after that's going to be changed. So how is that protein going to work as compared to the original? So sometimes you'll see some analogies where they compare it to 3-letter words. So if the normal sequence is see, the, dog, run, and I insert a letter, okay, or sorry, delete, and I get rid of that second E, everything's going to move over. And so instead of having a sentence with meaning, see the dog run, I have set head ogre un. Does that make any sense? Does that have meaning? Not really, which kind of means protein don't work right, as my dad would say. Insertion, if I insert another E right here, C et ido gluten. Still makes zero sense, right? Same thing with your body. You make that protein, your body's like, what the heck do I do with this? Thing don't work right. So here is a summary slide that has the normal, silent missense, nonsense, and a frameshift mutation, specifically a deletion. So I'll maybe pause this, spend some time with it, and then you're going to do some more stations. So let me know if you have questions. Okay, bye.